Hey everyone, this is video number 6 for the immune system. For the next 3 videos, I decided to break them down according to 3 main types of lymphocytes we've been talking about. And to talk about for each of them, what happens when they're activated by a foreign antigen, and what is the process that comes about after activation. What is the response, the immune response of each of these different types of lymphocytes? I'm going to start with B cells, and then I'll go through helper T cells and cytotoxic T cells. B cells in this video, and T cells in the next 2 videos. So let's take a look at sort of the outline for all 3 videos, which is shown here. And I showed the slide in the last video, and we talked about the first 4 steps in that video. Formation of lymphocytes, maturation, when they're mature, they get transported, and they perform immune surveillance. So in the next 3 videos, I'm going to be talking about these. What happens in immune surveillance when they're activated by a foreign antigen? What is clonal expansion? What are the effector responses that happen in an immune response? And what are memory cells? Okay, so that's the general outline for the next 3 videos. And this slide is just to remind you where we ended off in the previous video. For mature lymphocytes, they are immunocompetent and self-tolerant. Immunocompetent means they're equipped with a specific antigen receptor. Each lymphocyte has a unique antigen receptor, and there are 100,000 of these for each cell. For a given cell, they're all identical in terms of what type of antigen they can bind to. Self-tolerant means that they only recognize foreign antigens and not self-antigens. For B cells, the receptor alone is going to function in activation. For T cells, there are 2 subtypes. They require a receptor, T cell receptor, and a co-receptor. Helper T cells have the CD4 co-receptor. Cytotoxic T cells have the CD8 co-receptor. All right, so we're going to focus in on B cells in this video, and what's happening in B cells in general is this. There will be some kind of antigen challenge, which means when a B cell is sitting in a lymphatic organ, let's say a lymph node, along comes a foreign antigen, which its antigen receptor has complementary shape to. It will bind, and that naive lymphocyte, meaning not yet exposed to its foreign antigen, now has been activated. Okay? So activation is going to occur in any of the secondary lymphatic organs or tissues. And this part of the slide is illustrating that an antigen in the bloodstream will be taken to the spleen. And that's where there will be immunocompetent lymphocytes. An antigen in the skin is going to be sucked up into the lymphatic vessels and eventually get to the lymph node. And there are lymphocytes there. Antigens coming in through the mucosal membranes are going to be exposed to lymphocytes in the tonsils or the malt. So that's kind of where the exposure will happen. Now, after an antigen binds to a B cell receptor, that's going to cause activation, part of which is this, clonal expansion. In clonal expansion, the cell that binds to the foreign antigen is going to be stimulated to divide. And now, instead of having just one cell with this specific antigen that recognizes the foreign antigen, you have a whole army, thousands of cells with the identical B cell receptor, or T cell receptor if it's happening in T cells. Okay? And so then they're going to do battle. So that's the effector response. And the effector response is now you have this expanded clone of cells, and they are going to carry out some immune response, which is going to slow down or eliminate the pathogen from the body. Okay, so the effector response is going to be specific. It's different for what a B cell does, what a helper T cell does, and what a cytotoxic T cell does. In addition to the effector response, all three lymphocyte types are going to form memory cells. So these are part of the original clone that came about during clonal expansion. They're not going to be involved in the effector response, so they're not going to be doing the battle with the pathogen, but they will be stored away in the body. And if you're exposed a second time or third, fourth, subsequent time to the same antigen, you now have more of these cells that would recognize that specific antigen, and you're going to mount a better response. Okay? So specifically what happens in an activation of a B cell, what is the effector response, and what are the memory cells that come about? So I prepared a short video to describe this. Take a look at the video and then I'll review it in the PowerPoint as well. Okay, when a lymphocyte is performing immune surveillance in a lymphatic organ, let's say in a lymph node, and B cells and T cells in the lymph node are subject to lymph passing through the node. And maybe they carry in a foreign antigen, maybe a bacterium or a virus or something dissolved in the lymph. And a lymphocyte is exposed to this antigen, and its receptor, B cell receptor or T cell receptor, binds to this antigen. Then that causes activation of the lymphocyte. And activation leads to a series of events, which is going to end up producing an immune response, which is to do battle with this foreign antigen, to destroy it, to neutralize it, to interfere with its ability to cause problems in the body. So I wanted to start that by talking how that works for B lymphocytes, for helper T lymphocytes, and for cytotoxic T lymphocytes. So in this video, let's start with B lymphocytes. And let's suppose there's a virus, which is this hexagonal structure here. And this is flowing through a lymph node, and you have in the germinal center of a lymph node nodule, you have a lot of B cells. So here are 3 different B cells. They're mature, immunocompetent B cells, which means they have antibodies. They have a B cell receptor in their membranes. Okay? There are 3 different B cells. They have different antigen receptors. So this virus, let's say, is detected by one B cell number 2 here. The B cell receptor on this is able to bind to this virus. It recognizes this foreign antigen, whereas there is no binding to the B cell receptors on cell number 3 and cell number 1. So what happens is B cell number 2 is going to be activated. It's going to go through a series of steps which are going to culminate in the humoral response to this foreign antigen. B cells are involved in the humoral response. So what happens upon binding of this foreign antigen to a B cell? So I've listed the steps over here, and this is B cell activation here. B cell activation involves the following. First of all, the B cell receptor binds to the foreign antigen. Second thing that happens is B cell number 2 is going to divide rapidly. You end up with a huge number of cells that have the same exact antigen receptor because they're all derived from this one. This one gets selected to divide. start out with just one, you end up with thousands of the same, basically the same cell. So that's called clonal expansion. This cell that recognizes the foreign antigen is selected to divide, and now you have an army of cells just like number 2 there, okay? This clonal expansion is aided by the T helper cells, and I'll talk about that when we talk about T helper cell and, activation. What happens to the clone of B cell number 2? Most of those cells are going to become effector cells. Some of them will become memory cells. So what is the difference between those two? An effector cell is when a B cell becomes what's called a plasma cell. It differentiates, it changes. And what a plasma cell does is it makes the same B cell receptor, but instead of sticking it in the plasma membrane of the cell, it secretes it. Guess what? These are antibody molecules. So the B cell receptor here is just an antibody molecule that's planted in the plasma membrane. When a plasma cell derived from this cell number 2 secretes these antibody molecules, they're going to have the same binding specificity, meaning they're going to be able to bind to other virus particles in the body, okay? So from the expanded clone of cell number 2 here, most of them are going to become effector cells, which differentiate into plasma cells, and basically they become huge antibody factories, pumping out large amounts of antibody into the lymph, into the blood, okay? Antibodies then, they can, you've expanded not only the number of cells, let's say 1 to 1,000, but for each cell that becomes an effector cell, it's going to pump out huge amounts. I don't even know the numbers, hundreds of thousands of antibody molecules. So you've expanded the army. The antibody molecules you can think of as tags. When an antibody latches on to a foreign antigen, that's a tag for that antigen to be destroyed. So you can think of that as the tagging system of the body. If you have an antibody attached to you, good luck. You're out of luck, I guess, put it that way. All right, so most of the clone here becomes effector cells that form plasma cells. Some of them become memory cells. What are memory cells? Memory cells are basically like this B cell. They don't differentiate, but they persist in lymphatic organs for many, many, many years. They do circulate around the body a little bit, but they can end up in multiple spots. And they're going to sit there in germinal centers inside of lymphatic nodules. And if you're exposed again to this same virus, whatever it is, then instead of having just one of these cells, you already have in your body many of these memory cells poised to respond. Okay? So that's what happens in B cell activation. Okay, B cell activation then, just to review real quick. So the BCR on a B cell receptor, antigen receptor, latches on to some foreign antigen. Now, one thing I didn't say in the video is that for B cells, the antigen can be in any form. It can be on the surface of a bacterium, it can be a virus particle, it can be an abnormal cell in the body, anything. It can be a protein that's dissolved in the lymph or the blood plasma. B cells are not picky whatsoever about the form that the antigen is in. As long as the antigen is capable of being bound by the B cell receptor, then that's going to lead to activation. Okay? What happens after it binds, I also didn't mention this in the video, but the B cell, when it binds the foreign antigen, it phagocytizes that receptor and antigen complex, internalizes it. And then what it does is it presents that to a helper T cell. And the helper T cell, one that recognizes that antigen, is then going to be activated, and it's going to stimulate the clonal expansion of the B cell. So I did mention that clonal expansion is aided by helper T cells. So just a little added detail there. So the second stimulation here is, this is the helper T cell releasing chemicals called cytokines, and these are going to stimulate the clonal expansion of the activated B cell. Okay, B cell activation binds its antigen, internalizes it, and then it's helped out by helper T cells to expand. Clonal expansion is going to lead to two fates, either differentiation to form plasma cells, and those are the antibody factories. Okay, so those are the effector cells. Or if they don't differentiate, they stay as memory B cells, and they're, you know, in your lymphatic organs for many years, ready to respond if you're exposed to the same antigen later on. Okay, and this slide is just summarizing everything we talked about so far. Here's an immune-competent but naive B cell, naive meaning hasn't yet been exposed to its antigen that it recognized. Until now, here comes a foreign antigen that it binds to. And so that's going to lead to activation. This is the first stimulation, binding to the antigen. And then the B cell is going to internalize this, and it's going to put it on its surface where it can interact with a helper T cell. Helper T cell is going to release cytokines, one of which is IL-4, interleukin 4. That's going to stimulate this B cell to undergo clonal expansion. Clonal expansion. So now you have a number of identical B cells. Some of them are just going to remain as memory cells. They won't differentiate. Most of them will differentiate into plasma cells that are going to secrete antibodies. And then antibodies are going to do the job of eliminating the foreign antigen. And we'll talk about how that works a little bit later. All right, so that's the situation for activation and the effector response of B cells. See ya.